in some fundamental operations that have been studied in signal neurons, certain kinds of calculations that we now know individual neurons can do, and what it all means. So I want to start with this slide, which shows an example of a neuron complex dendritic tree been built with a fluorescent dye, and it's imaged at high resolution using a two-photon imaging system. And so on this image neuron, it's possible to see out in the dendrite these very, very fine structures and spines. They're of order a micron in dimension. So this technique is very precise. See in every time. The compartmental modeling is a technique that allows you to build a computational model of that dendritic tree, taking into account all of that complexity. So nowadays there are systems that can reveal these structures and take that data and break each portion of the dendrite into a single little tube and represent the electrical properties of the tube and concatenate them all together and build up a model of a complex dendritic tree, generate the equations automatically, numerically integrate the equations automatically, and simulate that neuron all from an image or model in a very quick way. And so this is how this all works. We're going to look at compartmental modeling again by considering just a single table of dendrite that is represented in the way that we talked about previously. These are axial resistances. This is a circuit element that represents the transmembrane resistance and the membrane capacitance. And so these units get concatenated end to end to build up the table. And if you zero in on this electrical circuit here, this represents what's going on in each one of these compartments in my figure. Circuit. This resistor here is the specific membrane resistance, the resistance of current flow through the surface area of the cylinder. This is a new element. It's a battery in series. The action of that neurotransmitter is to increase the permeability of the membrane. And so that's modeled as an increase in that membrane conductance. And that event, the increased membrane conductance, allows ions to flow into and out of the cell. And the direction of that current flow is determined by the value of this battery here, which represents the reversal potential of whatever ions are permeable at this synapse. So you know about reversal potentials. You've learned about them before. So this is a expanded view of this electrical circuit element. And so we can, if we label voltages at each node in this way, we can apply Kirchhoff's current law yet again at each node. So the current flowing from this node to this node through this resistor is given by this expression here. The current flowing out of this node is given by, flowing into this node in the other direction is this current here. Maybe there's an applied current, maybe there's an electrode at this dendritic location and current is being applied to the neuron, so that's what the capital I term represents. And so these three terms are this current plus this current plus this current, right? So those are the currents flowing into that node. And Ohm's law says that the currents entering the node must be balancing the currents leaving the node. So that current is a current through the membrane capacitance. That's this term here. And then there's this term. And I've been a little clumsy in my notation on this term. There really should be a current flowing through this resistor in parallel with this resistor, but I haven't written that. Instead,
I've ignored this conductance, this resistor, and I've only written an expression for the current flowing through the synaptic conductance. So there should actually be an additional added term here to this value of conductance. It would be the value of this conductor, but I was cheating. I was trying to get this done quickly, so I haven't written that down. This term here therefore represents this synaptic conductance, and so the current through that synaptic conductance is the voltage at this node minus the value of the battery, the reversal potential, times that synaptic conductance. So we're thinking of two kinds of inputs in this neuron: a synaptic conductance, this value of G, and applied currents. And so we can go through every node in this cable and write an equation like this. And then there are equations that are applied at the end of the cable. Let's assume this is a finite length cable. So there's an equation for the node, the first node over here, and the last node over there. All right? So you know how to do that. It's just Kirchhoff's current law. And so we're going to manipulate these equations and put them in a more useful form. And to do that, we're going to define a membrane potential V that is the difference between, I should have said V star, which is the difference between the membrane potential and the reversal potential. So that's V star, and we'll just take that term and plug it in to the equations in the previous slide, and we get these equations here. So I'm just rewriting them in terms of a new voltage relative to whatever the resting potential may be. So V star is 0 if transmembrane potential is equal to that reversal potential. So we're just recalibrating the voltage range here. All right, so now we are going to construct some vectors and matrices. Let this vector have as elements the V star values for all k nodes in that cable. So that's what we're calling this vector. This vector has as elements whatever the injected currents are at each node. Maybe they're 0, maybe they're non-zero, but here they are, and we're calling that vector U. And then we're going to define a matrix, a k by k matrix B. It's a diagonal matrix, and the diagonal terms are going to be 1 over the capacitor values for each of the nodes. So if we make these substitutions, we can look back at the form of, I'm sorry. We're going to define another matrix A. And so this matrix A is going to have this structure here, where the rows are defined accordingly and the C's are just various membrane capacitance values. So armed with this vector V star, U, matrix B, the matrix A, and these parameter definitions, we can go back and rewrite these equations in matrix form. So this is easy to do. You can figure it out after lecture. And so we get a matrix form for the underlying differential equations that govern the time rate of change of voltage in that dendritic cable, and it has this form. The vector V star, the time rate of change of the elements of vector V star is equal to this matrix A times V star plus the matrix B times U. Remember, U was a vector with all of the current inputs as elements. A is the state transition matrix. This is a linear time invariant system. The elements of A are constant. So it's a constant coefficient k by k state transition matrix. And for a cable, the matrix A has a particular structure. For an unbranched cable, there will only be elements of A along the diagonal and just off the diagonal. That's because you pick a node in that cable, it's got a left-hand and a right-hand neighbor. And so if that's the case, then the state transition matrix A will have this tridiagonal structure. Now in general, for if the cable is not simple, if it's not unbranched, if it is branched, then A gets filled in. The elements of A get filled in. It's not a tridiagonal matrix. And the way A fills in depends on how you would sweep through a dendritic tree and number each of the nodes that you're considering, each of the little compartments of membrane that you're considering. So if you use one numbering scheme, certain elements of A will get filled in. If you chose another numbering scheme, different elements of A would get filled in. But that's a detail. The big picture is, even for a complex branch neuron, the underlying equations will have this general form. And so you can solve them on computer. You can use MATLAB, for example, because you've got the velocity field defined. It's the stuff on the right-hand side. So you can numerically integrate these equations and, for example, apply any pattern of synaptic inputs to the dendrites and simulate what happens when you vary that synaptic conductance G, and solve for voltage anywhere in the dendritic tree and understand how this neuron responds to different kinds of inputs through numerical simulation. And furthermore, as I said with respect to the first slide, since we have imaging methods that can give us very high resolution views of entire dendritic trees now, we can computationally decompose that dendritic tree into a bunch of interconnected sub-elements. Knowing the length and the diameter of each of those sub-elements, we can compute the specific axial resistance, the specific membrane resistances, and we can get all the terms that we need in this equation to numerically simulate a given real neuron's branching pattern and how it responds to different kinds of synaptic inputs or different kinds of applied currents. All right. We could let the synaptic conductance changes vary in time. We could let the applied currents vary in time. We can still solve for voltage everywhere in a complex dendritic tree. We could also simulate the effects of applying a current and leaving that current on for a long time and look at the steady state distribution of voltages in this dendritic tree in the same way that we figured out how to analytically solve that problem using Rawls cable theory. And so we can do it computationally as well. And so we want to look at the nature of that solution. And so in the steady state, dV dt is 0, voltages aren't changing, so the left-hand side is 0, and we're left with these terms, and so 0 is equal to the negative of A inverse times the matrix B times the vector U. Remember, U has as elements the applied currents. So we've got a voltage, a steady state voltage, equals all of this stuff times a bunch of currents. So this term here, so this is Ohm's law, E equals IR. Right? This matrix gives what are known as transfer resistances. We'll call them K sub ij. The ijth element of this matrix here relates current applied at node I to voltage produced at node J. So we have the elements of this matrix telling us the relationship between applied current and voltage everywhere in the dendritic tree. So these are known as transfer resistances. This is where they come from. So you see how they come from the underlying system of differential equations defining a compartmental model. And we'll return to this concept of transfer resistances later on and use them. But this is how you get them when you're actually working on and solving a compartmental modeling problem. Okay, so that's really all there is to compartmental modeling. It's conceptually very simple, very powerful. You give me a good quality image of a neuron and its dendritic trees. I can use this approach to generate a computational model of that neuron, and I can study how that neuron then processes information. And the first kind of information processing that we're going to now think about is what has been called neural integration. And so what are some general concepts underlying the way in which neurons combine synaptic inputs within their dendritic tree to compute and analyze those inputs. Okay, so to think about this problem, we're going to again define a simplified dendrite as a single cable attached to a cell body. We're going to consider injecting current at one location in this dendritic cable. And we're also going to consider the effects of applying a synaptic input at that location on the dendritic cable. The synaptic input will be modeled in this way. There will be a synaptic conductance in series with a battery. That battery again is the reversal potential that provides the driving force on the ions that move through this particular ligand-gated ion channel that is the target of a neurotransmitter. And that produces some current, and then we'll consider that that current is applied at this point in the dendritic cable, and in this way we'll model the effect of changing a dendritic conductance, increasing the conductance, allowing current to flow into the neuron as would happen with real synaptic activation, and we'll look at the properties of the neuron. So, all right. So, what we would like to do is to derive an expression that relates the effect of applying a synaptic conductance change, a steady, maintained synaptic conductance change at some location in the dendritic tree to the voltage produced at that location and the voltage produced at other locations by that synaptic input. So that's what we're going for here. And so remember that when a synapse, a receptor, binds a neurotransmitter, the action of that neurotransmitter is to increase membrane conductance. That is the value G. And that increases permeability to whatever ions move through that channel. And those ions have reversal potential E. And V sub I at location I is the voltage at location I. And so the effective current applied by this by turning on the synaptic inductance is this expression here, just Ohm's law. Okay? So the voltage at that location is equal to the applied current times the input resistance. So remember, I previously said K sub Ij are transfer resistances, meaning they are the relationship between current and voltage when current is applied at location I and we want to compute voltage generated at location J. Well, here the subscripts are both I. And so this is the voltage, this is the input resistance looking into the dendrite at the site where the synaptic conductance change is applied, right? There's some current generated, we've got that equation. So that current times the input resistance equals the voltage at that same location, right? So if you bring down this expression for current and plug it in, we can then solve for voltage at location I. It has this form. So let's just pause here and think about the voltage that is produced at a particular location in a dendrite by changing the membrane conductance, mimicking synaptic input. What you see is that as G gets large, the larger we make the synaptic input, the larger this term becomes, and this expression approaches 1. Okay? So what that in turn says is the largest voltage you can produce by applying a synaptic conductance change at that site is the voltage E. It's the reversal potential. Now this is different from the case of injecting current into a dendrite. If you inject current, the larger you make the current, the larger you make the voltage at the site of current injection. But when you're thinking of your input as a synaptic conductance change, you don't get that kind of linear relationship. Instead, you get a saturating nonlinear relationship between the applied conductance change and voltage. It's this relationship here. It's a sigmoidal function. Because as G gets big, that expression goes to 1. So you're limited in how much charge you can inject into that neuron at that site by changing conductance. And therefore you're limited at how much voltage you can produce at that site. So this is a nonlinear relationship between conductance and the voltage that it generates. Whereas if we were thinking of a synapse as simply injecting some current into that location, there's a linear relationship between that current and the produced voltage given by Ohm's law, and we could make the voltage as large as we want by injecting more current. And so what this real relationship is telling us is that you can only make the voltage so large, and this effect is known as synaptic saturation. No matter how much we increase membrane permeability, we can never make voltage at that location larger than the reversal potential E. It's called synaptic saturation. You can only do so much at that site. Okay, so let's continue with our analysis here. We've got an expression for the voltage at the applied conductance change. So the voltage produced by that change at the soma is given by the current that is injected at the site where the synaptic conductance change is located times the transfer resistance KIS, right? Because that transfer resistance relates current applied at location I to voltage produced at location S. Here S is indicating the soma of our model neuron. And so the ratio of these two voltages is just going to be the input resistance where the conductance change is applied divided by this transfer resistance. So that's the voltage. So we can rearrange terms and write an expression for voltage at the soma, and then we can plug in voltage at location I produced by the synaptic conductance change, and we get our final expression, right? So we know that out on the dendrite, the biggest depolarization that can be produced by the synapse is the potential E, the reversal potential. So that's, and therefore the biggest potential that we can produce at the soma is that value E times K sub IS divided by K sub II. This is the transfer resistance between the site of the applied conductance change in the soma. This is the input resistance at the site of the applied conductance change. So that ratio now scales E. And that's the biggest response we can get at the soma. And as we increase the applied conductance change, again we get this saturating behavior. Voltage at the soma will grow, but as G gets larger, this term will asymptotically approach 1. And so this is now the maximum potential that we can get at the soma. Now, therefore, knowing these transfer resistances is useful because you can use them to compute voltage distributions within a dendritic tree. So again, this property is called synaptic saturation, and this shows you what these curves look like. So here is a plot of the term of the term that scales the reversal potential in that previous expression. So if we plot that term, this expression as a function of the base 10 log of G times KII, that's what that scaling function looks like. And here we see the effect of synaptic saturation. So why is this significant? Okay, let's think about this one. All right. So suppose that G is takes on a large value, a large enough value, so that G times KII, the input resistance, is 100. Okay? So we're operating way out here. A log of 100 is 2, and so this term is near 1. The voltage produced at that site where this conductance is applied is going to be its maximum value. It's going to be the reversal potential E. Okay. Now, let's imagine applying the same amount of total conductance. In other words, G times KII equals 100, but we're going to apply it at 10 different locations in the dendritic tree, and at each site, G times that site's KII will be 10. So we've got 10 of them. It's the same amount of conductance. It's just going to be spatially distributed, not all applied at one point. So if GKII is now 10, log of 10 is 1. So this scaling term is about 0.9. So at each site where we apply one of these synaptic inputs, the voltage at the soma will be 0.9 times this term. But imagine we've got 10 of them, and they're at 10 different locations in the dendritic tree, and they each produce this amount of depolarization at the soma. So the effects are going to combine in a linear way. So at the soma, we now have a maximum potential of 9 times this term. So what's the consequence of that? If we take a large amount of conductance and apply it at one location in a dendritic tree, through the effects of synaptic saturation, we can't produce a lot of voltage elsewhere in the dendritic tree. But if we split that same amount of total conductance across different sites within a dendritic tree that are electrically isolated from one another, we can get a big amplification of the response. In the example I'm working out, we're getting 10 times the response by distributing the inputs. Yes?
That linear summation, that's because of superposition?
It's because of superposition, and we're assuming that the sites where we're applying these inputs are far away enough from each other that they aren't interacting electrically together. And so it's an approximate number. It's approximately 9 times as large. But this is the point about neural integration, dendritic integration. If a dendritic tree is sufficiently large that it supports many different locations that can function independently of one another, you can produce a much larger voltage response at the soma by distributing inputs, the same amount of input, the same net amount of input, rather than by focusing all of those inputs in one location or 2 locations. So bigger response when you distribute inputs. That's because of this phenomena called synaptic saturation. And its advantage to a neuron is that if the dendritic tree is large enough to support electrically distinct regions, the same amount of conductance more effectively will drive that neuron produce a larger voltage response at the soma. Maybe that's important computationally. Maybe distributing inputs in this way helps assure a larger depolarization in the soma and helps assure that a neuron fires. So more reliable firing because a larger depolarization, superthreshold depolarization, is produced at the soma. So again, integration, and the idea is that spatially distributing inputs is more effective than confining those inputs to the same spatial location. All right, so that's neural computation 1, and so these are examples of that calculated from real neurons. This is work done by Christoph Koch and Tommaso Poggio when Christoph was a postdoc at MIT with Tommaso Poggio. This is a retinal ganglion cell. It's a particular type of ganglion cell known as an alpha ganglion cell. Because it's a cell in the retina, and the retina is flat. The dendritic trees are flat, so they liked studying these cells because they could look at them in the microscope, project an image of the neuron onto tracing paper, literally tracing paper, trace out the dendritic tree, and they knew they were tracing an intrinsically 2D object. So they were looking at a two-dimensional dendritic tree, projecting it onto a two-dimensional piece of paper, rather than looking at a three-dimensional dendritic tree, projecting onto a 2D piece of paper. So by tracing out the dendrites, they were able to, offline, so to speak, decompose the dendritic tree into small subcompartments of known size and figure out the electrical parameters for those compartments and build compartmental models and study the electrical properties of the dendritic tree. And so here's what they found. While we won't go into the details of how they did this calculation, they are claiming through numerical calculations that each of these shaded regions are electrically distinct regions of the dendritic tree. That is to say, what happens in this shaded region isn't affected by and doesn't affect what happens in this shaded region. And so one can now imagine that since there are a fair number of electrically isolated regions, that if we were to distribute inputs not all together in one region, but divide them up across regions, that it would be, we could design a way in which to make this neuron fire very reliably. That is to say, those inputs would produce a larger depolarization at the spike generation point near the axon hillock. Okay, so here's another kind of ganglion cell. Whoops, let's go back. See the scale bar, it's 100 microns. Here's the scale bar, it's 100 microns again. So this is a much more compact dendritic tree. And the electrically isolated regions are much smaller, much more compact. There's still a fair number of them, but they're much more confined. So this whole neuron is small and electrically compact. In fact, the size of the whole neuron is about the size of one of those shaded regions for the previous alpha ganglion cell. And so while there are a few electrically isolated regions, they're small, and if we distribute inputs across this dendritic tree, they tend to land in regions of the dendritic tree that are not electrically isolated from one another. So this neuron is sufficiently compact that it may suffer from an increased degree of synaptic saturation with respect to inputs. And so the bottom line is that neurons like this one, with a fair number of large, spatially segregated, electrically distinct regions, are neurons that do integration well. They can combine inputs well, whereas neurons with fewer electrically isolated regions or more compact ones are less able to do that. And so, because those neurons suffer more from the effects of synaptic saturation. Okay, so that's the first concept I wanted to present about how neurons, in a very general idea, are able to combine together inputs or do it less well so. So that's called neuronal integration. That's one thing you need to know from this lecture. The second thing you need to know from this lecture is another kind of computation that appears over and over again in neural signal processing. It's called coincidence detection, and coincidence detection is an example of an AND operator. So here's a real neuron. It's from the nucleus magnocellularis in bird, I think. It's essentially a bipolar neuron. You see the soma here, and there's two dendritic branches coming off that soma. And in general, they don't branch all that much. So what's interesting about this neuron is that it's in a part of the auditory system. And And so this neuron is receiving synaptic inputs from the cochlea on one side of the brain, and they are all segregated onto one dendritic branch. And then the other dendritic branch gets inputs from the cochlea on the other side of the head. And so there's dual innervation of this neuron with inputs from opposing sides of the head landing on these two different dendritic branches. So there's ipsilateral and contralateral input. Okay, so I want to skip to the movie, then I'll return to the other slides. So here are the bipolar neurons living in the brainstem, and they are receiving inputs from the contralateral cochlea and the ipsilateral cochlea. And so this delay line hypothesis is a very famous hypothesis about how sound localization is done in the horizontal plane. So suppose, and this is the creature that this was first studied in is barn owl, right? Barn owls are very good at locating mice in the dark. And they have entire neural systems specialized to help them locate where a sound source is in the horizontal plane. So suppose mouse is over here rustling in the grass. That sound reaches this ear before it gets to this ear. We know that the key encoded information telling us about where in this direction that mouse is located is interaural time delay. The sound will get here before it gets here. We know that neurons, central neurons, like these bipolar neurons, encode interaural time delay. That is, there's a whole bunch of these neurons arranged together, and at any particular location in the brainstem, that group of neurons is sensitive to a particular interaural time delay. And so if that neuron, or that small group of neuron fires, the barn owl knows what the interaural time delay is, and therefore knows if the sound got here first or here and here second, and what was the delay, and therefore the barn owl can, in principle, compute that's where the mouse is. And it can do this with very high resolution. So this has been studied by putting barn owls in anechoic chambers and presenting a click, not a mouse, a click, to this ear and a click to this ear with a controlled interaural time delay, and watching where they look. And they make very precise head movements to the right location corresponding to that particular interaural time delay. So interaural time delay tells us where in the horizontal plane that sound source is located. So this guy here had a hypothesis about how it was done. So let's watch this movie. I hope, oh yeah, it's actually working. That's nice. Okay, let me stop it. You see what happened? That sound wave came, so that's the sound wave, that's the representation of the sound wave. It's going to arrive at both ears simultaneously because we're looking straight ahead and the sound source is straight ahead. So interaural time delay is 0. So the idea here is that the sound is encoded in primary auditory nerve fiber firings projecting from the cochlea. These nerve fibers project to the anteroventral cochlear nucleus. They innervate bushy cells in the anteroventral cochlear nucleus. They fire every time one of these projection neurons fires. They're very reliable signalers, and so we get an action potential in the primary auditory nerve firing that bushy cell, causing it to propagate an action potential and activate synaptic input on this bipolar neuron. So let's continue watching the movie and see what happens. And what I want you to watch for is which neuron lights up to represent it firing an action potential. Okay, so let's continue. Whoa, 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 that was stunning. Okay. Okay, the movie's running again. Okay, there's the sound. Watch what's happening. Okay, that neuron lights up. It's doing it again. Watch it again. Second neuron from the top. Different travel times. Oh, slow-mo. Okay. All right. So different path lengths. And so the different travel times correspond to interoral time delays. So for an interoral time delay of 0, neuron number 2 fires. Now keep watching. Sound's coming from one side. And it takes longer to get to the other side. So we've introduced an interoral time delay. So now these action potentials are going to arrive with different delays, and they're going to travel along these paths with different delays, and so the neuron that fires encodes that interoral time delay. It was neuron number 6, I think. So this is called the time delay hypothesis. And the reason that neuron fires is that due to the differences in travel time along these pathways versus this pathway, each of these neurons will encode a different interoral time delay, and the signals from the two sides must arise coincidentally to cause that neuron to fire. If that happens, that neuron is able to fire and encode interoral time delay. So what that neuron is doing is coincidence detection. The inputs are arriving at the same time, and it's also implementing an AND operator, because these other neurons don't fire because their inputs are arriving at different times. So that means to be an AND operator, receiving one input alone cannot make the neuron fire. Both inputs arriving simultaneously must be the only way the neuron can fire. If that is true, the neuron can implement an AND operation. Input 1 and input 2 must be present to fire. And it's implementing coincidence detection. So let's go back and think about how we can analyze this quantitatively, in this case using Rawl cable theory. Okay. So you had a problem like this. So here's our bipolar neuron model. A soma is represented simply by resistance. We're only going to do a steady state analysis of this problem. Here are the two dendritic cables of our bipolar neuron model. So let's imagine supplying inputs on both sides at the same distances from the soma. So we're not varying input location in this tree and that tree. Let's just have them at the same distance from the soma, and considers what happens when those inputs are very close to the soma and when we move them out away from the soma. So when the inputs are very close to the soma, intuitively you would think that one input might be able to effectively activate the soma if it's very close to it, right? What's the reasoning for that? Imagine applying a steady current at this location. It's close to the soma. There's not a lot of opportunity for decay of membrane potential from this site to the soma. So that one input can increase voltage at the soma effectively. And if it does so enough to be above threshold, then one input alone can electrically activate and fire this neuron. That's not an AND gate. We don't want that. We want this neuron to only fire when two inputs arrive together. So how do we figure out how to build this AND gate? Now imagine moving those inputs further and further away from the soma. Any single input, as the further away it is positioned, will produce a smaller and smaller potential at the soma. There will be some distance at which that input will not be able to produce an above threshold response for a given value of applied current, that is, at the soma. So that's the distance, the location in the dendritic tree where we may be able to begin building AND gates. That's where we want to apply our input to implement the AND function. Because if we provide two of them, one on each dendrite, together they may produce a response at the soma that can fire an action potential. Okay. But now we move them further and further away from the soma. And now there's going to be a distance that is sufficiently large, there may be, if the dendritic tree is large enough, that the two inputs together, one on each of the dendrites, can never be able to raise potential at the soma enough to make it fire an action potential. So there is some intermediate range here where if we apply inputs here and here, we can implement an AND gate. If we're further away from that, we won't get any response at the soma. And if we're closer than that, every input will evoke a response. That's not an AND gate. So this is the thinking, the kind of a general thinking about the principles behind building an AND gate. And what's neat is that we've actually got here a pretty reasonable model of those cells that I was showing you. They're just bipolar cells. They have two dendrites coming off the soma. The ipsilateral and contralateral inputs are segregated onto the two separate sites. Makes sense that that neuron's implementing an AND gate. Okay. I have 3.5 minutes left. We're going to talk about the AND NOT operator. Now, why do I care about the AND NOT operator? Well, Koch and Poggio showed that neurons can implement an AND NOT operation. Who cares? Well, if you can build a neuronal AND gate and you can build a neuronal AND NOT gate, you can build any kind of neuronal logic circuit. Any logical circuit can be built from a composition of AND NOT gates. They are capable of universal computation. So the field got pretty excited by the fact that here is a way that neurons can be implementing universal computations composed of and NOT operators. What's the idea? The core idea is a concept known as shunting inhibition. So inhibition, an inhibitory input, increases membrane conductance to ionic species that causes membrane hyperpolarization. Shunting inhibitory input increases conductance to an ionic species whose resting potential is near that of the cell. So if this reversal potential were 0, right? Increasing this conductance produces no response at this location in the dendritic tree if the neuron is sitting at a membrane potential of 0 millivolts, right? Produces no response alone. And so you wouldn't see the effect of just the inhibitory input. It doesn't produce a voltage response. But what it does is it kind of opens a hole in the membrane. So if current is injected out here and that current is moving towards the soma, turning on this conductance can shunt it out and divert it from reaching the soma. So this is called shunting inhibition. So the way they built an AND NOT operator intuitively is to use this shunting inhibition. So if you get an input here, and if the conductance values are kind of right, the input here ablates the response to this input. So you get a response if this input is on and this one is not. I hope that makes intuitive sense. This input alone will not change membrane potential. You don't see any response to it. But if this conductance is large, it doesn't by itself produce a response anywhere in the dendritic tree, but it's able to divert charge from reaching other regions of the dendrite and the soma. It's shunting inhibition. So that's the intuitive idea of how to build an AND NOT operator. And so neurons can now implement AND NOT operators and build any logical operation within their dendritic tree. So I'm out of time, and so I prove this. I prove how it works. You can read through the derivation. It's good to know. And we will resume with another final topic on whatever day, I guess Monday or Wednesday, whenever I next lecture. All right, thanks.